J'espère que tout le monde m'entend, parce que j'entends un bruit bizarre. Voilà. Donc je suis Jérémy Robert, le consul général de France à New York. J'ai pris mes fonctions dernièrement, très récemment, en fait, il y a 3 semaines. Et donc je suis très heureux de vous retrouver, Ah, okay, sorry, sorry, in French. So after this word in French, let me just switch to English. So I'm Jérémy Robert, the Consul General of France in New York. I joined my mission very recently. I started 3 weeks ago. I replaced Anne-Claire Legendre, whom you know very well. And I'm very happy to meet you virtually. A bit frustrated because usually these conferences take place in the consulate, but I'm very happy to meet you through Zoom today, and hopefully very soon directly to start the new season of the conference 934. The conferences 934, as you know, they are usually they aim at targeting a theme which is very linked to the current situation, and which is also linked to the situation in France or in the US, or sometimes in France and in the US. And this is the case of the theme today, which is an issue, the research for vaccination against COVID-19, an issue which is global, and an issue which is very important for both our countries, France and the US, because our two countries have suffered dramatically, and we this week we have reached the terrific figure of 1 million people dead because of the pandemic, with more than 30 million people infected. So this is we thought that it was very important to start this new season of the conferences with this very important issue, which is the race for vaccination and the international cooperation, the international scientific cooperation to find vaccination. We know that there are different projects for the research and the development of vaccination. And we know, as you may know, France has also, at the political level, with our European Union partners, we have taken a political initiative to make sure and to ensure that once the vaccination will be developed, different vaccination will be developed, there will be a clear process to distribute the vaccination and to make it available to everybody worldwide, including in the most vulnerable countries and among the most vulnerable population. And this is with a target to make this vaccination as a global good for humanity. And President Macron recently, when he addressed the General Assembly of the United Nations, put this priority very clear once again. I'm just here to introduce our two very prominent researchers who are going to give our views on this topic today. We are very lucky and very privileged to have two very important experts at the forefront of the efforts to combat COVID-19. I would like first to introduce Dr. Simon Wain-Hobson. He's a professor at Institut Pasteur. I'm not going to introduce Institut Pasteur. You all know the quality, the excellency of the work of Institut Pasteur, who has the mission for so many decades, and I think about 100 years, to combat disease and by research and development of cure or vaccination. So the Institut Pasteur is an institution whom we are very proud of, a French institution, which has also a huge network worldwide to a scientific network to develop the medicine or the vaccination, and also a network to act on the ground in Asia and mostly in Africa also. Among this key task for Institut Pasteur, Dr. Simon Wain-Hobson is also board chair of the Foundation for Vaccine Research in Washington, D.C., which is a nonprofit working on, as the name mentioned it, on vaccination research. And in the 1990s, he has published the genetic map of the HIV virus, and he continues to research on the origin of the HIV. He's now contributing, is a key virologist who is key to contribute to the global efforts to develop a vaccine for COVID-19. I would like also to introduce Dr. Marie-Paule Kieny. She's a director of research at Inserm. Inserm is the Institut National de Santé de Recherche Médicale. It's a public institution in France, a governmental institution, public institution, and it's a leading European research institution on medical field and scientific field. It's a focus on human health, with also the priority to also find ways for treatment and for innovations. Inserm has played a role in key medical advances, such as gene therapy, in vitro fertilization, and radiotherapy for cancer. Marie-Paule is in charge of a priority research program of Inserm on antibiotic resistance that France initiated in 2019. And she holds many titles in various international research, health and development organizations, such as the board of directors of the Human Vaccine Project in the US and Fondation Mérieux in France. I have really the honor to welcome these two prestigious guests today to address us on this very important topic. And I would like now to give the floor to Anpresh. Anpresh is our scientific attaché based in Boston, and she kindly accepted to facilitate the discussion today. Anne, I give you the floor, and have a good conference.
Thank you very much, Monsieur le Consul Général, and welcome to our guests, Professor Wain-Hobson and Dr. Kieny. Welcome also to our audience. We're welcoming your questions through the webinar, and I would encourage you to send them through the chat window that you can open on the Zoom session, or during the Q&A session, you may also raise your hand so that we can let you ask your question directly. Or if there is an opportunity, you will be able to unmute directly. And but please do that only when we're going to have the Q&A session, and stick to the chat window right now. So we just heard the numbers, and I'm, as always, numbed when I'm listening to them. 1 million lives claimed, 200,000 of which were living in the US, and as you all know, New York City being hit so hard. So we're dealing with a very contagious virus that wreaks havoc on the human body, targeting a whole set of different organs, lungs, the kidneys, liver, blood vessels, etc. We heard a lot of different symptoms, and we're just beginning to understand the virus long-term effects. So Professor Wain-Hobson, you're a renowned virologist, and you have a long-standing record of working on HIV. And you recently shifted, like many of your colleagues, your work to the global race against this coronavirus. And thank you very much for that. And I would like also to extend my many thanks to your colleagues, to all of the scientists who are working 24/7 on the coronavirus. So could you please, Professor Wain-Hobson, share with us some of the lessons that you have drawn from your work on HIV, how it compares to the fight against the coronavirus?
Thank you very much. And yes, Marie-Paule is going to give you the breakdown on the vaccination. So I decided to step back a little bit and try and tell your listeners how unusual this is. And I cut my teeth in HIV back in 1984. And we were the first to publish the genetic blueprint of the virus. And this, the number of déjà vu I'm having and déjà entendu is amazing. If only to start with HIV, everyone said, where did this thing come from? So immediately we had, yes, it's the CIA. And then they said, no, it's the KGB. And the simple answer to that was, I knew nobody that was smart enough to make a virus like HIV. And conspiracy theories are peddled around, as you know, and they are very abundant when there's little information, and they get wiped out when we have more data. This virus is a natural virus. How can I say it? There is no hand of man in the genome, not that I can see, and most of my colleagues feel the same. But our technology is so powerful that it might be in our lifetime, we might see a microbe that has been engineered by man, and we have to be very careful. It's probably 20 years away, 30 years away. That's how powerful. Now, we've made tremendous progress fighting viruses and microbes, but who would have thought that a microbe could close down New York and reduce Fifth Avenue to an eerily quiet, to quote a friend of mine in New York. You talk about 1 million, but can we have some context? Yes, that's 1 million in 8 months. That's 1.5 per year, at least. Right now, tuberculosis TB is knocking out and killing 1.7 million a year. Fewer people are dying of AIDS than we will see of COVID in this year. And malaria, the numbers are even smaller, about 440,000 deaths. So this is a big one. It's 9 on the Richter scale of microbes. And we have to admit the brutality, the emergence of this virus showed that it was, that pandemics are and can be unanticipated. You know the old adage, prevention is better than cure. But let's do some number crunches. You know, when last year, we used to talk in numbers, economic numbers, and we were using billions, tens of billions, hundreds of billions. Now, people are talking of trillions. The economic cost of this for the airline industry, hotels, restaurants, and all the attendant societal problems. Actually, we didn't have that with HIV. HIV was it discombobulated the world, but it never had this. And just to put things in context, trillions of dollars are mentioned. The cost of making any vaccine has never been more than 10 billion. So, you know, we have to think the prevention, we have to keep track of viruses and maybe make vaccines in anticipation. Going back to HIV, well, I was young at the time, so I use it as a reference. But COVID is HIV times 10. I was just before I came on, I looked and did a Google, and the word COVID is used 16 times more than HIV. HIV has been going since 1981, and in the few months of COVID, with the web, it's more referenced. It's warp speed. We did the sequence of the HIV genome in 3 weeks, and people were saying, "How did you do that?" Now it takes less than 24 hours. We talk about vaccines, and Marie-Paule is very well placed to give you the summary. But your listeners might not know that she will be talking about a phase 3 trial. This is a phase 3 trial in less than 8 months since the virus was identified. And this is absolutely outrageous for a scientist, anyone who has any reference. This is words like wild, stunning come to mind, and it's an impressive, very impressive performance. And we have to see that all this is being done in collaboration with the FDA and the appropriate authorities. And it's good to know that these people can adjust very fast when it's necessary. And it's a tribute to so many people, not just science, but in the regulatory areas that Marie-Paule will be telling you about, not just one vaccine, but several that are in phase 3, and it's absolutely stunning. As a virologist, when we have a new virus, everyone is saying, well, where does it come from? And you look for the closest virus to try and learn. And as you know, we had the SARS virus in 2003, and the number of deaths, I think, were less than 1,000. And it turned out not to be the right model. It didn't, apart from being a coronavirus, it didn't help us much in terms of pathology, nor did this other disease that's provoked by a camel coronavirus. I'll give you just one example. From the earlier SARS epidemic and the MERS that hit hard South Korea, pregnant women had a death rate of about 40%. So when SARS came along, it just happened that my daughter-in-law was pregnant. And so we were scanning the web and wondering, you know, what's going to happen and how could we protect her? And it turns out the SARS-2 is not an issue for pregnant women. So you can see with just that one example how difficult it is to extrapolate physiologic pathology and the societal consequences from data. But as we had nothing else, it was our reference point. And it reminds us once again the problems of doing science. And I like the little image that science is like playing with a chess master, a grandmaster. But you're playing with a grandmaster and you don't even know the rules. You're learning them as you go along. And that's what we do in science. And it's amazing in the present context that we will be talking about vaccines in less than 1 year. And this is fantastic. But science and medicine do need time. And as with HIV, and we're not going to get all aspects of this virus sorted out in 1 year. It's going to take time, and have a really efficient vaccine is going to take time. There's only one human virus we've eradicated by vaccination, that was smallpox. Polio is pretty beat, but we're not quite there. Another aspect which is going to transform the way we scientists and infectious disease people see things. This is a respiratory disease, and one of the big problems is it's very hard to biopsy the lung. You can do what we call a bronchial lavage, which is rather painful. HIV was, by comparison, simple. You just had a blood sample, or you could biopsy a lymph node, and you could see what was going on. But people are becoming creative, and we're going to see some fascinating science, and it's going to revolutionize disease immunology and the way we do infectious disease research for these respiratory viruses. And this is, well, I see it from HIV, the downstream effects of the investment in COVID is going to revolutionize the way we do respiratory virus disease. So there's going to be some wonderful science emerging in the next 10 years. And just one, I would like to close with one simple remark. I was young when I was working on HIV. I'm a baby boomer. And as you know, the risk factors for COVID are diabetes, overweight, cardiovascular disease. And I escaped all these. I dodged nature's microbes and other arrows. And as a baby boomer, accumulated privilege and property. We could call ourselves a lucky post-war generation. And microbes are always there to get you. Because I'm over 65, I find myself in a risk group. And yet I want to say I'm healthy, I feel fine, but I have to be particularly careful because I'm in the risk group. You can never escape microbes, and one should never think it possible. But people from Pasteur on have been doing their level best, and it's going to happen, and I think I should stop there, but it's a very, very impressive achievement. It's going to take time. There's a long road ahead, and we're not going to wipe it out fast. But as Marie-Paule will tell you, there's some very interesting work coming along the road, so I think I should stop there. Thank you, Anne.
Thank you very much, Professor Hopson, Wain-Hobson, for sharing your insights as a baby boomer. And we have a question, and I guess the question is a little bit related to the fact that there is it's kind of a theory, but I mean, it's debated of the and you talked about it, of the nature of the virus, and was it maybe fabricated, or has it been recombined with other viruses? So Dominique Lallemand is asking us if there is any HIV element in the COVID SARS-2 genetic sequence, and could you comment on that?
Yes, that was fake news. Okay, to use a trendy term. Yes, this how can I say, people do like to talk about themselves, and this was a number of scientists unfortunately got involved in this. There's no data for it, and it's sad, but the people who did those analyses didn't really they didn't have a good understanding of the methods they were using. So it's out, it's finished, time over. Game over. Not true. It's a natural virus. It happens to come from China, but could I point out that the last flu pandemic came from Mexico? We didn't call it Mexico flu. This MERS virus came from Saudi Arabian camels and moved and caused great damage in South Korea. Viruses move and emerge. This is the way it is. And this one happens to be from China. HIV was out of Africa. There we are. This is the way things go.
So thank you. It's kind of interesting. I think that this question of origin of the viruses and the way it's spreading, and I'm just going to ask you a question, a personal question. How do you see that? I mean, how do you think we could boost research on you know, the study of viruses coming from I don't know, bats or other animals? You were talking about the camel, and so what can we do? And then we have another question in between that just came in, and it's, do we know yet if COVID will be a dormant virus in the people infected like HIV or herpes? So two kind of questions that if you could just answer.
Well, can I do the last one? I mean, the good news about COVID, unlike HIV and herpes, is that those two viruses, herpes, we used to say, we have a black joke in microbiology, herpes virus infections are forever. And same with HIV. This is a virus that will come and goes, and your immune system will wipe it out, just as it will wipe out a polio virus or a flu virus or a common cold virus. So it's not going to reside. It might linger after your initial primary infection for maybe several months. We now know from very sensitive testing that all the virus is not eliminated in a matter of days, but I would say you're not infectious after your antibodies are kicked in, and the virus will slowly be eliminated. You won't be infectious once your antibodies are up. As to where it comes from, yes, there's a huge amount of work being done, how viruses cross over to humans. And lots of people studying it. The NIH has put a lot of investment in this. But can I just point out that no one predicted this virus and the way this virus behaves. I think we must get out of this mindset that we can control everything, we can anticipate everything. We're in a world, we're living with microbes. Microbes see us as prey, and we're just not going to get everything. It's just not possible. And we must stop thinking. We've got to be more philosophical and say that we're going to miss a few things, but when it comes, we have to mobilize. And the mobilization of COVID is phenomenal in the scientific world, phenomenal. I've lived HIV, but this is just fantastic.
Thank you. So we're just a little bit late, and we could come to the last question a little bit later. I would like to give the mic to Marie-Paule Kieny. Dr. Kieny, could you please, maybe you would like to share some slides with us and about the vaccine and the research that's been done on it. You have to unmute, Dr. Kieny.
My audio, is it better now?
Yes,
good.
It is.
So let me now put in full screen. Come on. There is a delay. You see it in full screen or not? Not yet. Coming.
Yeah, we can see it pretty well.
No, I think it will come in full screen. Okay, and so I have to forget that. Good. Is it okay? Mm-hmm. Fine. So I thought I would give you a status of where we stand right now. So you will see that I have adapted from illustration that have come out in very popular US journals, Financial Times, Washington Post, the New York Times. And this is because actually I think that if there is something really where I think that the US is really much better than we are in France. This is about putting things complex into something that can be understandable by a non-expert. And this is why, you know, going through a museum, for example, in the US is such a joy. So I have taken these slides from these three journals. In addition, they are updated every couple of days, and I have adapted them to my where I wanted to use them. So this first slide you can see, and this is changing every day, where are we now in terms of a vaccine pipeline? So currently there are no vaccine which is approved to go really downstream for use in the general population. But there are already 5 vaccines which have been approved for early or limited use. And I will go through all of these. Now, the phase 3, which is the stage where vaccines are tested for efficacy in man, you now have 11 vaccines. In phase 2, 14. In phase 1, the first step in man, 27, and 200 more in preclinical development, either in the lab or in animal models. So, and this is changing every day. So this gives you an idea of the numbers. They may have even changed today, but this is the type of order of magnitude. So now, what does it mean? So this is the process for development. You have the first step is actually the step where you develop a prototype. So this is essentially in the lab, you construct with bricks and mortars, you construct something that could become a vaccine in the future, a prototype. After this, what you do is that you have to test it in animals. That's the preclinical testing. You test it in mice, in non-human primates, in ferrets, in hamsters, to see whether it induces immune response and whether it protects in these models. And if everything's fine, then you go into phase 1 first-in-man clinical trial, which is given usually in a few dozen people. If it's still good, looks fine, then you go into a phase 2 trial, or you expand your phase 1 to a phase 2, as they do in COVID, and you test in a few hundred people. If it still looks good, then you go into phase 3 efficacy trial, where you look at efficacy and you vaccinate for COVID, for example, about 15,000 people, you have 15,000 controls, and you look for efficacy. So what I have put between parentheses here is early or limited approval, which is not the case usually for normal vaccine, and which exists for COVID. And finally, you go to approval. So what are the type of vaccine? Quite often we talk about platforms. What does it mean? you have different types of vaccines. So the usual one that you see down in the bottom are the viruses in terms of vaccine against viruses. They can be either attenuated, so they are weakened, or they can be killed with usually chemical agents. So this is the classical types of vaccine, and many vaccines currently in use in man are of these two types. Then you have over there on the right, you have what is called a subunit vaccine. So this is made only of a fraction of a pathogen. It's either a peptide or a recombinant protein, or even this recombinant protein can be formulated as a virus-like particle in a nanoparticle of lipids. So there are also quite a number of vaccines for humans which are based on subunit vaccine. The next one, which is coming closer in terms of development to these days, are made of viral vectors. So this means you take a shuttle, a vector, a virus, which is not pathogenic for humans, and you add in this virus something from the coronavirus or another one, and they can be either non-replicative or replicative. And there are many of the COVID vaccine candidates which are made on this prototype. Finally, you have the newest vaccine, which are just made of genetic material. Could it be either RNA or DNA? And this is really a novelty for COVID, because there is currently no human vaccine based on DNA or RNA, whereas there are vaccines made out of viral vectors, and there are plenty made of the other types of vaccines. So what are the vaccines which are currently approved for limited use? So there's one vaccine out of Russia, which is made out of two viral vectors, adenovirus, prime, a first dose of a recombinant AD26, then a second dose of an AD5 boost. There are also 4 inactivated vaccines, all coming from China, one from a private company, Sinovac, and the others, we have 3 here, Sinovac, and these 2 which are made of inactivated vaccine, and the CanSino vaccine, which is an AD5. How were they approved? Well, they were approved based on their safety in man, in small numbers, the immunogenicity, so they make antibodies in small numbers of humans, and in general, they have been proven to protect in animals. So what are the vaccines which are currently in phase 3 efficacy testing? So 11 of them, only one which is non-specific, in parentheses on the right bottom, which is BCG, because there is some ideas that BCG, the vaccine against tuberculosis, because it induces a non-specific response, could help protect against COVID or against SARS-CoV-2 infection. And there is currently a phase 3 trial to try to demonstrate this concept. So what are these vaccines in efficacy trials? So there are 3 which are inactivated vaccine that you can see here, the 3 of them from China. There is one which is a subunit vaccine made from Novavax. Then you have 4 of them which are based on vectors, adenovirus-based vectored vaccine. So AstraZeneca, Oxford, that I'm sure you hear about in the US. Then you have Johnson & Johnson, or Janssen, according to whether you see them from Europe or from the US. And then the one that I already mentioned, which have an approval, CanSino, and the Russian product. You also have 2 mRNA-based vaccines developed by Moderna and by BioNTech Pfizer. So where are these vaccines undergoing phase 3 trials? So of course, there's not much which is happening in Europe in terms of testing, because now recently the number of cases in Europe has also started to grow. The Oxford AstraZeneca vaccine is tested in the UK. Apart from that, you can see many vaccines are tested for efficacy in the US here. And otherwise, they are tested in area where there is a high prevalence and a large scale circulation SARS-CoV-2 vaccine, because if you want to test the vaccine, you need to have, and if you want to do it quickly, you need to have a high level circulation of a virus. So how much was spent so far on COVID vaccine? So this is increasing all the time, but as you can see, clearly the US is the country which has spent the highest on the development of COVID vaccine. And if we were to be able to access COVID vaccine in early 2021, certainly that would be mainly due to the US investment. Now you can see that the European Union and the European Commission is also investing quite a lot in funding services in helping scale up of production and also to secure supply. The UK government itself is funding nearly as much as the whole of the European Union. Then a lot by the Bill and Melinda Gates Foundation for development, and a new initiative coming out of the European Commission, but called also in his wishes by the French President Emmanuel Macron. He has also started to put quite a lot of money on the table to purchase vaccine, especially for low and middle income countries. And you can see here some funding from the government of Brazil. So usually, how long does it take to develop vaccines? So you can see here, it can go from everything between 34 years for chickenpox. That's, I would say, an extreme, because usually it's quicker than that. And before coronavirus, of 7 years for polio. Now, will we have a vaccine in 1 year for coronavirus? It's, I would say we can be reasonably positive that we should have some vaccines, not only one, but some vaccine, which confer some protection against COVID-19 by the end of the year. So there's still a lot of unknown. Protection, yes, but protection against what? The disease, so the pulmonary manifestation, only the severe cases, or even no disease at all? Or will it be protection against infection? Will it be protection against transmission? Of course, it would seem very important to protect against transmission if we want to protect the community. How much is protection? 50%? More? There seems to be an agreement that to accept that the vaccine is worth using, it should protect at least 50%. But then people say, well, the lower confidence bar, the lower bar of the confidence interval can be anything between 30% and 0. So how is this possible? I think if you can take two representations. Either you take the Russian dolls, you know what it is, and you have all these Russian dolls are just put in one. And what you can do is that you can open the largest and the second and the fourth, and you put them one near the other. This is how it is normally for the development of a vaccine, that you do everything one after the other, and then it takes a long time. You can also put all the dolls together, and then you have the impression that you do less, but actually you do the same, but they are all done at the same time, in the same major doll. You can also use the image of an accordion. If you use an accordion to play music, then if you pull it, it's very large. If then you put everything together, all the foils are still there, but they are all on a stack. So what's happening to develop COVID vaccine is that all the steps are done, but instead of doing them one after the other, they are done in parallel. And this is how it is possible to condense this time into 1 year. We don't have a vaccine right now. Let's make sure that I make it clear. We don't know if we will have a vaccine by the end of the year, but one can be reasonably positive that it might be possible that indeed by the end of the year there will be a vaccine authorized for emergency use, and that we hope in the weeks and months after that the population will have access to this vaccine. Thanks a lot. I'm stopping the share of slides, and I'll be happy to hear your comments, if any.
Thank you, Dr. Kieny, for this inspiring slides. We have a number of questions. One has just emerged a little bit before your talk, but I think it's very relevant also to the vaccine. What do we know about the longevity of the antibodies? And I mean, in a way that's also pertinent for the vaccine.
Well, you know, in terms of antibodies generated by immunization, what we can see is that normally in different vaccines, it's variable. For some vaccines, you have a situation which is very stable for years. Some others, you can see that shortly after the peak of immune response, it comes down. For COVID, SARS-CoV-2, we still don't know, because the results that we have seen are all for the time being very shortly after the vaccination, at the peak of vaccination, if I may say. And because everything was so quick, we will have to wait 6 months to see whether the antibodies last for 6 months or not.
Thank you. You were talking about the threshold for the vaccine acceptance in the US, which is like the FDA has put it at 50%. And Marc Cherpion is asking us if this is also the accepted threshold for Europe.
I think in Europe and at the WHO also, and in the US, 50% seems to be a magic number. Where how would I say, some controversy arises as to how low can the lower bound of a confidence interval be. Let me explain. You know, the efficacy will be determined, the point efficacy actually, with very little number. So the idea is that when there are 150 cases in the group, which is made of vaccinated and controlled people, the authorities then will look at how many are in the vaccinated group and how many are in the placebo group, in the control group. So these are still so small numbers. And it could be that, you know, because of the imprecision that you have, because it's a small group, you will be able to say, yes, my point estimate is 50. But actually, statistically, I have a 90% chance of not being wrong by saying that my true efficacy is actually between 30 and 70. So that would still be acceptable. But would it be acceptable to say, I have 95% chances of being right, saying that actually my true efficacy is between 0 and 70? So what does it mean? If it could be 0, potentially it's very low. And so there is discussion about whether it would be acceptable to say that say that we want to deploy a vaccine where the actual efficacy would be between 0 and 70.
Thank you. So I'm trying to a little bit go back to the questions. One of the questions was from Christian Roche, and he was asking us about which type of vaccine is the flu vaccine. I think it was more when you were showing the slide on the different kind of vaccine that have been made right now. Could you comment on this one?
Is it about flu vaccine, you mean?
Yes.
Did I hear that?
Which type is the flu vaccine?
So the traditional flu vaccine, the most common, is an inactivated vaccine. So this is a virus which is grown in cell culture or in eggs usually, and then killed with a chemical. Now, there has been in the US in particular for quite a number of years an attenuated vaccine, which is the one made by MedImmune. And more recently, there has been a vaccine which is of a subunit type, a recombinant one, was also accepted for use in humans. And this is now sold by Institut Pasteur.
Thank you. So Margaret Marjin is telling us that she's glad to know that the BCG may protect from SARS and COVID. I mean, she is commenting and saying that's good for all the baby boomers. And she was asking about the interferon, type 1 interferon publication that just was released in Science very recently. Could you also comment on that, and any recommendation for the general population?
Well, you know, the trial to see whether BCG could protect through an unspecific mechanism, could be through interferon, by the way, is not to rely on a previous vaccination, but to do a new vaccination, a second vaccination with BCG. I think the trial is in healthcare workers because they are more exposed, and to see whether it protects them to a certain degree against infection with SARS-CoV-2. For the time being, we don't know that. Well, I have seen, indeed, there has been some news and a publication showing that potentially there is, no, it's not potentially, there are hard data showing that there is a difference in the severity of a disease in some people which have some mutation in interferon gene. There is also some evidence, although not complete, that inhaled interferon can have an effect also as a cure, or at least to blunt the severity of COVID-19. So all these needs to be further evaluated in carefully designed clinical trials, and I think we should know more in a couple of months.
Right. I did understand also that it might be used also for a screening, the level of interferon to try to predict who would have a severe disease, severe COVID symptoms.
Yeah, and this is linked to the fact that there seems to be some differences in interferon gene for people who have severe disease or not. But it's really very early. So this is a very interesting discovery, but it's too early to know how much practical application it will have.
Thank you. Another question from Pascal Dadoun, who's asking if we're looking for a yearly vaccine like the flu vaccine or a one-shot. I guess it's also...
we don't know. For the time being, you know, we don't know how much protection we will have, and we know even less how long protection will last.
Okay. And Dominique Lallemand is asking us if you can clarify the timeline, availability of a vaccine before the end of the year 2020, or 12 months, like end of March 2021 or later?
It may depend where. As you know, the US government has put a lot of money on the table to secure very early access to a lot of vaccine for the US population. So I guess that vaccine will be available very early in the US. In Europe, we'll have to see. I understand that we will have the first supply of vaccine at the end of the year, but more towards the first and the second trimester. So, but you know, this is something that we start also to discuss with the vaccine scientific committee. All the data that we will have on safety will be on 15 for each of the vaccine in around 1,500 people who have been vaccinated, and the safety data that we will have by the end of the year will be for a few months only. So the question is, in terms of safety, what kind of jump can you do? Can you really rely on safety data on 15,000 people to vaccinate 50 million? Or would it seem more prudent to go from 15,000 to 1 million, 1.5 million, and wait a few weeks, a couple of months, to see whether indeed this is completely safe before you really vaccinate 100 million? So there's a lot of discussion which are starting right now to see how much you can immediately scale up and how much you should do this in steps.
Thank you. I think we have some time for the more general Q&A session. I think we have something like 8 minutes left, and if there are some other questions for Simon Wain-Hobson. So I see that a few just came in. Dr. Wain-Hobson from Marguerite Mangin, any chance to find a vaccine for HIV? So we're back to HIV. Thanks to all those sorts related approach.
HIV is our black hole. We've done everything, incredible performance on drugs, epidemiology, transforming the science, everything. It's our black hole, and it weighs on all of us. People are still working on it, but no one dares make a prediction anymore. But could I point out, sadly, that we don't have a good TB vaccine, and we don't have a good malaria vaccine, and we don't have a good HCV vaccine, and we have lots of vaccines which we would love to have, and many of them have not been done through a lack of investment, but that's another issue.
So thank you, Simon. There is a question from Philippe who's asking, how do you see the availability of several possible vaccines and the choice of one from the other? I guess it's also related to the risk that one can take with new type of vaccine, like the mRNA vaccine, and some people are more comfortable with the recombinant one. So what would you be your comment on that?
Well, I don't know. We have to see. You know, for the time being, we start to have a good idea of what is called the reactogenicity of this vaccine, which is, you know, how much fever do we have? What % of people have fever immediately after vaccination? What percentage has a sore arm? So this starts to be quite well known, but on rarer side effects, we don't know, and it is only when a larger number of people will have been immunized that we will have absolute certainty about what's the safety profile.
Thank you. Dominique Lallemand is also asking about the political pressure in the US to move fast for the vaccine. And I guess this is also related to the safety issues.
Well, you know, I can only say that I think that science and politics don't go well together, and that in agreement with a scientist in the US, I think that you know, we should follow a scientific rationale and a scientific view of how to develop vaccine and how quickly it is safe to put them in use, and not be influenced by politics. But, you know, this being said, who am I to say something like that?
So a few more questions just came in, and I'm sorry if it's, you know, we're going back and forth and safety, politics, and the timeline, but Valérie is asking us if you know how the vaccine will be used. Will the vaccination be for prevention or treatment? And will it be used on general population, people at risk first, or what kind of people at risk? People who are sick or have been sick?
Well, vaccines in general are used to prevent. Yeah, of course, they are also development of vaccines for therapeutic vaccine against cancer, but the vaccine will essentially be used to prevent. So this is in people who are not infected yet, which is to avoid that they get infected with SARS-CoV-2.
Dominique is asking also if either of you could comment on the progress on the treatment.
Do you want to do this, Simon, or do you want me to do it?
It's pretty simple. When you take treatment, the big thing is the toxicity. As a rule of thumb, all your listeners know that you take these tablets, and these are what we call small molecules. And there's a rough rule of thumb that every 10 that go into the clinic, maybe only 1 comes out. And this is because of the side effects. And these side effects you really cannot predict. For us, one drug, it could be a liver, for another, it could be kidney. And this is a trial and error. And if you run too fast on those, then you will hurt patients. Tremendous progress, interesting molecules coming through. Some are incredibly efficient, but it's going to take time. With the vaccine products that Marie-Paule has been mentioning, many of these have been tried and tested for other vaccines. And so we know quite a bit about the possible side effects. But for small molecules, every time it's a new game and we cannot extrapolate. So there again, sadly, please give us time, but the progress is stunning.
Yeah, well, for the time being, there is no antiviral which is really doing the job of eliminating the virus or of diminishing the mortality of sick people. This is not to say that it will not happen. The only molecule which is really very efficacious, has been proven to be efficacious in a trial in the UK, is dexamethasone. This is a very old drug, and it's been proven to reduce mortality in severe patients by 30%. Now, what we have to see is a new class of drugs, which are the monoclonal antibodies, are now being tested, and there is hope that the efficacy of these drugs will be higher than what has been tested so far.
Thank you. We have 2 more questions, but we're right at the end of unfortunately, of our meeting, and I don't think we have so much time to go through them. So before leaving, I would like to thank you, Professor Wain-Hobson and Dr. Kieny, for sharing your thoughts and answering our questions. Thank you also to the attendees for participating and for your insightful questions. And finally, I would like also to thank our Consul General, Jeremy Robert, as well as the team of the French Consulate in New York for having organized such a wonderful meeting. Thank you very much.
Thank you so much, and take care, everybody.
Take care.
Bye-bye.
Thanks to all of you.
Thank you. Bye.